Hello, I'm a Brain Research UK and Royal College of Surgeons of England Research Fellow and a clinical academic trainee in neurosurgery in Cardiff. It's a privilege to share our work today on genome instability in gliomas as part of the London Global Cancer Week this year. Glioblastoma is the commonest primary high-grade brain cancer, and despite maximal treatment, overall survival remains a dismal 5% at 5 years for our patients. Of all solid organ cancers, these patients also have the greatest mean years of potential life lost that they would have otherwise lived in good health. In many solid organ tumors over the last few decades, the overall survival of patients has improved as newer therapies have emerged. The bottom pink line shows that for brain cancer, this is still not the case, and we need to find urgent newer treatments to try and improve outcomes. Crucial to improving outcomes and finding newer treatments for our patients is understanding how glioblastoma develops and grows in the first place. In our cells, chromosomes carry packed genetic information within DNA, and chromosomes have caps at their ends called telomeres. These shorten each time a cell divides. However, as shown on the diagram on the left, in a cancer cell, telomeres can control their lengths, and the cell can potentially divide indefinitely. This is a key common step in the formation and growth of many cancers, and as shown on the right, labeled are all the genes that we know to be implicated in what makes a brain cancer grow. In green are those highlighted that are related to telomere function, so we know that telomeres are crucial to having high-grade brain cancers develop. We just don't quite know why yet. Our lab has expertise in telomere biology, and one way of knowing cancerous tissue from normal tissue is looking at their telomere lengths. We have shown this with other groups in bowel cancer, breast cancer, and blood cancers, where normal tissues have longer telomeres as shown in green at the top of the pages, compared to cancerous tissue which has shorter telomeres as shown in red. Even as a tumor grows, we have seen that telomere lengths continually erode compared to normal tissue lengths expected. And we have shown the same with some exciting pilot data in 100 patients with glioblastoma. As shown in the survival curves on this page in red and blue, patients with shorter telomeres at the bottom or an unmethylated MGMT status at the top, which is the current best available molecular biomarker for this brain cancer, do significantly worse in their overall survival compared to patients with longer telomeres or methylated MGMT status in blue. This suggests that telomere length can be an accurate and novel biomarker for overall survival in our patients, which is exciting. To study this further, I'll be using a pioneering technique which our lab has developed to accurately measure telomere lengths on a high throughput scale for large cancer cohorts, like patients with glioblastoma. We are fortunate to have internationally leading expertise on this technique within Cardiff, and my data collection will be generously supported by the Royal College of Surgeons Research Fellowship. The overall aim of this project is to better predict the survival of our patients and responses to newer targeted therapies for GBM. I will start by validating our data in 100 more patients with GBM and analyze their telomere lengths. I will also look for telomere fusions in these patients as another hallmark of genome instability. Later on down the line, we will create a bespoke 3D cell model in the laboratory of our patients' brain cancers, and we will be able to try newer targeted therapies on these models to be able to predict how patients will do depending on if they have shorter or longer telomere lengths and what their DNA signature looks like. This project represents novel and promising work in hopefully being able to better predict survival outcomes in our patients with GBM and also their response to newer potential targeted therapies for their personalized medicine. I'm privileged to have excellent supervision and national collaborators, and I'm grateful to be able to draw upon the international expertise within the Telomere Lab in Cardiff and be supported by a clinical academic programme within Wales. We hope that in the future we can bring you back some exciting results from this project as well, and we are grateful for this platform to be able to share our work. Thank you.